From Microbe TV, this is TWiEVO. This week in evolution, episode number 118, recorded on September 10, 2025. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Great to be with you from LD Lab Studios. And we're back in the game a little bit quicker than usual. Our kind of schedules conspired so that we decided to hop on. And we've got some fresh science, which I think that the strike while the iron is hot, as they say.
Yeah, well, we have colliding schedules, so
Right.
sometimes it's hard to find the time, but we always do it once a month, right, Nels?
Yeah. And so we're here a week or two early, but the trains are running ahead of schedule. Might be another way of
Welcome, Barb Mack, our sole moderator today. I am guilty of
Great.
posting this only an hour, 2 hours ago. I'm very sorry. I've got some
okay.
travel, and
Yeah.
I've been going crazy this week trying to prepare for it.
It's great.
So it may just be you and me and a couple of people, Nels, but it'll be recorded and people can listen to it. There's not going to be an office hours for the next 3 weeks because I am traveling and can't do it on the road very easily. Although, so today I can't because I'm on a train.
Yeah.
And next week I'm in Greece. The timing is all fakakta. I don't think I can do that. And the following week I'm at a textbook retreat. I really have to be hanging out with my co-authors, not running off and doing office hours. So this will give you your live stream science fix.
There we go.
For the next couple of weeks. So we have moderating today, Barb Mack, UK. Thank you for coming. And Les Fabi, thank you so much. And I apologize again to everyone who's here for the late posting of this episode. I have to bring my recording equipment to Boston. I'm doing a podcast there. And every time I have to pack it, it's like, where's this? Where's that? Where did it go? We come back from a trip, I put everything away, and then I forget where I put it.
A lot of moving pieces there. I can vouch for that. When we did our on the road in vivo live stream of TWiEVO 100 down at the Science Mill in Johnson City, Texas. Wow. Yeah, you're not bluffing. It's a big production to
a
take
lot of equipment
to
go around. So let's see. Androot is here. Welcome, Androot.
Welcome.
Oh, Dejeen from Ethiopia.
Fantastic. Welcome.
Wow. Ethiopia. I don't know if we ever had someone from Ethiopia before.
Yeah, new
That's
dot
cool.
on the map. Yeah.
And look who's here, Bob LD.
Oh, there's a familiar face.
The Salish Sea. What's the Salish Sea? Do you know?
Yeah, I do. So he's coming from the Pacific Northwest, Puget Sound area, aka this part of the Salish Sea.
Nice. I bet it's cool and cloudy there.
Yeah, well, let's get the weather report.
Oh, who knows? It could be sunny. I don't know.
Summer's pretty great.
He's here to check how you're doing, Nels. Make sure you're...
That's right. This is my new kind of family reunion. Is everyone, if they want to track me down, show up on a TWiEVO live stream and we can compare notes.
Tom Steinberg is here from Eugene, Oregon.
Fantastic. Welcome, Tom.
Glad to see you. So I am going to Boston on a train. I'm going to hang out at a meeting with a lot of people. Yeah. I'm going to on Sunday, I'm going to Greece on a plane, and I'm taking a train for 4 hours, and then I'm going to hang out at a meeting with a lot of people. So what am I doing to protect myself? Because this is COVID season, right?
Indeed. Yeah.
So, here, I'm going to tell you, Vanity Nutrition is here too. Thank you so much.
Oh,
all these moderators, when I screw up and don't post it on time, we still have it. So I am going to wear a mask most of the time. I bought this fancy mask, which has an insert that has copper in it.
Oh, wow. You're going all in. Good idea.
Because copper inactivates viruses. It's supposed to be good for long trips. It's comfortable. It's reusable. It's $40 for a mask. It's not a throwaway. So I'm going to try that. Plus, I got vaccinated last Saturday, and
Good call.
it's not 2 weeks yet, but it is really, in my view, a boost. So it should be faster than 2
Yep.
weeks, and I hope that's good enough to protect me because I am over 65, and so I'm at higher risk for severe flu or COVID. I have RSV vaccines already, and that's a one and done. You don't need to repeat that as far as we know.
Beautiful. Yeah. Good idea.
So that's what I'm doing. Michael
call.
Schmidt. Hello, Dr. Schmidt, who will be doing an episode of TWiEVO in my absence.
Oh, fantastic.
Yes. So, Michael, I got a copper insert on this mask. So it has an outer layer, it's black, and then there's another layer that goes inside, and they give you two of them, and they're copper infused. So
good timing. I think I saw, there's another lineage of SARS-2 that is now dominating. I think it's the 11th dominant lineage coming at it from an evolutionary standpoint that has swept through. So there's new variation out there. Great time to take these exact precautions that you're describing.
Copper, the wonder transition element. And no wonder when you renovate a house, the plumbers come and cut out all the copper pipes. They resell them. So Vanity got spike vax, kicked my butt. So I got the high dose flu for old people and the Moderna mRNA, the new formulation. I did a little sore arm for 24 hours, and that's it. So I'm just Mr. Superman.
Super immunity. I will say locally, I'll draw two connections here. First, we're waiting on the SARS-2 vaccines, which are delayed because of all of the monkeying around nonsense that's happening.
Yeah.
And secondly, as it relates to copper, I'm out the window of LD Lab Studios. I can see the largest open pit copper mine in the world.
Wow.
Kennecott mine across the Ochre Mountains. And so some of that copper that you're going to be using to filter out the virus particles could have come from sort of my backyard here.
Could be.
Yeah.
So here, Richard is from Northern California, COVID trending higher. Yeah, it's been going up in many places for a while. It's a very strange epidemiological pattern. We have two peaks now in the year. We have one about now, and it'll probably subside, and then around December it will go back up again. I don't really understand why. Usually we have one peak of flu during the winter, so something's going on we don't know about, and people will need to find out what's
still
going
learning
on.
Agreed. Still learning as we go.
That the danger is to assume that all viruses are the same.
Agreed.
Just like we should never assume that any two people are the same because they're different. They have different DNAs, right? And so to say this vaccine works for everyone in the same way is not right.
Yeah. Well, and it's cutting in a couple directions here. I'd say, no people are the same. The number of viruses out there outnumber people by orders of beyond orders of magnitude, and so the variation that we are only barely glimpsing at is there's a lot to learn or a lot to know.
Okay, Michael Schmidt has a wonderful factoid. 80% of the copper ever taken from Mother Earth is still in use. Can we say that about petrochemicals? Absolutely not. Yeah. It's friggin' carbon dioxide, right?
Yeah.
Oh my gosh, that's just amazing. Yeah, as the plumbers, they just come in and cut it out because they're going to resell it, and it's going to be reused. The copper is bloody expensive. Yep. Here we have Miloš from the sad police state of Serbia. Oh, I'm so sorry. Riot police and gendarmerie inside the University of Novi Sad campus. I'm sorry to hear that. 27C. Next summer, I'll be going to Czech Republic.
Oh, great. Yeah.
For the coronavirus meeting. That should be a lot of fun. Let's see what the weather is here. It is in Chelsea, 20C and cloudy. Yeah, it's now in the 60s, That's it. Summer went boom, immediately gone.
Same here. We've been dipping down into the low 60s or even the 50s at night. Warming up still, though, in the day.
Andrew says maybe it's just something as simple as the equilibrium not having settled yet. Flu has had a long time to stake out into an equilibrium. It could be, yes. COVID's only been around 5 years, right?
Yep.
SARS-CoV-2. So, yeah, it's feeling itself trying to decide. Of course, viruses don't decide anything. It's all about evolution and selection. And this program is where you can learn all about that.
That's right. So, Vincent, speaking of learning, we've got a paper to talk about, but
Well, before we do that, I
yeah,
think you know what you're going to mention. Go ahead.
yeah,
Go ahead.
I wanted to, first offer my condolences on the passing of David Baltimore, your postdoc mentor, an absolute giant in the field of virology and beyond, in the field in science in general and beyond. And so I wanted to thank you, Vincent, for posting TWiEVO 100, which was an interview 15 years ago with David Baltimore. I didn't get a chance to meet him, but seeing that interview, I think, opened a really inspiring window into a little bit of who he was.
So as soon as I heard he had died, I went and listened to it, and it was actually really good. Oh my gosh! We asked good questions, and he had great answers. And then I store all the old episodes on hard drives, right?
Mm-hmm.
Two hard drives, redundancy, and same with our chromosomes right now.
That's right. The diplades. We'll get to the genetics and the evolution very soon.
So it was on the third disc.
Yeah.
And I opened the folder, and there is a video that I had taken of David, but I never published it because it was just him. And back then we used Skype, and you couldn't record video, and I only had one camera. I couldn't record myself. I didn't know. So I just released the audio. So I released the video of him sitting there, but it works because you can see him thinking. And I put in this cool fade at the end where he says goodbye and he just fades away, and then the empty chair is sitting there.
I noticed that.
I like that. When I
agree.
Did that, it just came out, and I said, oh, this works. This works really well.
Yep. No, so if anyone within TWiEVO earshot, please do yourself a favor and find that link on the Microbe TV, either at the YouTube site, at the TWiEVO website. It's an important one. And I think, 15 years on, the exact ideas you were talking about, obviously are on the menu today and echoing on in terms of.
That's incredible. He was pretty prescient about talking about all the different things which are relevant today, for sure.
Yeah.
And he said very interesting things. Like he said, science is really hard, and it gets harder to do. It doesn't get easier. It gets harder to find good people. It gets harder to get funding. It gets harder to keep a lab. And it's all true.
Yeah.
Especially when people are fighting you, it gets even harder. Right?
That's right.
Go ahead.
Well, I was just going to say your question to him about sort of given the headwinds, he was talking about sort of just over the course of a career, right? As you go from grad student in academic science, from grad student to postdoc to running a lab and beyond. But the question you asked him about, well, what kind of, where are you coming from in terms, or is it the viruses that you're, that motivate you to do the work, or is it that the viruses can teach you about the molecular biology that sort of underwrites the way life works? And he answered it was the latter. And then, seeing that, I think, what struck me was, or I think what was really in evidence from your interview, which also included Rich Condit and Alan Dove, was just sort of David Baltimore's liberal arts approach to science. I don't think it's a coincidence that he got his undergrad degree at Swarthmore, given sort of the range of the way, not only, the details of the molecular biology of the science, sort of tuning in on viruses as a way of getting access to that. But then what does that mean for things like biosecurity, ethics, how it fits into the bigger picture? I thought your questions and conversation about the Asilomar meeting. So, back in the 70s, the safety issues around all of this new technology, recombinant technology. And in particular, Vincent, I was really moved or inspired by his ideas about how, that kind of conversation that he helped to, to put together or to organize around the Asilomar recombinant biosafety meeting is not really possible today, or even 15 years ago, given the difficulty of just finding the space for scientists to sort of weigh things, come to some sort of a consensus, and then, sort of present an idea to the bigger public. And I think that really that kind of hit close to home for me, as we, and I think as you and I have been talking about over the years about the difference of the speed of science, which is quite slow, versus the speed of politics or the speed of, the news cycle, et cetera. And yeah, kind of remembering that wisdom has been out there for decades, and how do we reclaim or claim it in today's situations? Yeah.
So Ramtin is here from Iran. Welcome.
Fantastic. Welcome.
Lovely to have you. Tom agrees that TWiEVO 100 is good, and Les also. Ramtin is an immunologist, have watched every episode and really enjoy it. Thank you very much. Really appreciate it. Yeah. There was a point in that TWiEVO 100 where you might notice I got up and walked across the camera. What I did was to go get a plaque assay plate. It was I had some in my office. I wanted to show, and that's what we had a little conversation about plaque assays. Yeah. Here's the episode that Barb Max put the link up for us.
Oh, thank you, Barb. Yeah,
get to it. That's great. Tom writes, the early TWiEVO Fauci interview was also very good. I recall the question, what keeps you up at night? Yeah, that was Rich's question. Oh my gosh, how prescient. That was pre-pandemic, of course, right?
Of course, yeah.
And in fact, when I was looking for this episode, I came across a picture of him sitting in his office. It's very, very cool. So yes, this is very interesting. Both David and Howard Temin went to Swarthmore.
Yeah.
And that's where David met Howard. Now, if you read this biography of David by Shane Crotty, Ahead of the Curve, okay, you will know that this is where David recognized that Howard was doing good science and he wanted to be his competitor. He talks about that in the book.
I love it. Yeah.
And so, Howard was the first to have an idea of how RNA tumor viruses transform cells. They made a DNA intermediate, and he spent 10 years looking for the enzyme. And then David spent 2 weeks doing experiments and found the enzyme.
Yeah,
he calls Temin. He says, Howard, we found it. We're going to publish it in Nature. I just wanted to let you know. And they end up publishing it back to back.
Exactly.
Which was a really, really civilized thing to do, right?
Yep.
And I always loved that. In fact, he tells that story at another TWiEVO that I did at Cold Spring Harbor for the 50th anniversary of reverse transcriptase discovery. I should pull some of these clips out and make a montage, because I think that's the way science should be. People should be considerate. But he recognized that Temin had been working on it for a long time, but he wasn't a biochemist, whereas David was a biochemist and he knew how to do enzyme assays, right?
Yeah, turn the crank.
But science is something where you all should join together and pool your expertise, not say, I did it first. That doesn't really matter in science. What matters is that you get it done. Right, Nels?
You get it done and what you care about. So I think if you sincerely care about learning more about the world around us, how it works at the microscopic or molecular level in this case, then how do you find that, and how do we get to that information faster? And it's not by sort of stepping on the backs of others, it's by lifting each other up. And so and I think maybe echoes a little bit on that idea of stepping back, looking at the bigger picture, taking your time to do things in a way that I think the long-term benefits of that approach to science is really undervalued. And so I think a great reminder of that as well. But Vincent, I wanted to ask you maybe, when you were a postdoc in the Baltimore lab working on the full molecular clone of poliovirus, I'm sure you've been sort of thinking back to those days, and what would you say kind of sticks with you about that time that you spent working with David, in his lab?
By the way, this is the book, Ahead of the Curve.
Great,
David Baltimore's Life and Science by Shane Crotty. Shane Crotty, of course, an amazing immunologist, but he wrote this, I think, for some kind of thesis many years ago, right? So actually, it's interesting because a reporter for, I think, Chemical and Engineering News called me to talk about David. And so it brings back a lot of thoughts, right? So I go to this lab, and I was scared to death because I come out of Peter Palazzi's lab with 4 people, and I go there. There's like 30 people,
Hmm.
and each and every one of them is friggin' brilliant. And I said, "Oh my God, what am I doing here? I just do not belong." Oops, sorry. I do not belong here. And
Yeah.
I was scared to death.
Mm.
And I remember that the first when I went for an interview, um, I didn't know where to go, and I met Bob Weinberg. I didn't know who he was either, but he said, "Can I help you?" I said, "I'm looking for David Baltimore." He said, "Do you have an appointment?" I said, "Yeah, I'm going." He said, "Well, follow me then."
That's great.
And
Yeah.
he was so nice. And then I get to
Mm.
the lab, and what I remember, first of all, everybody was welcoming. It was, Steve Goff was there at the time, Fred
Oh,
Alt, who
yeah,
ended up
yeah,
coming to Columbia,
yeah,
and many other
yeah,
smart people, Al Bothwell. And I could go to any number of people for help, and they knew how to do everything.
yeah. Mm.
David didn't have to be around for that. He was just
Gotcha.
around
Yeah.
for the bigger thought pictures, right?
Yep. Yep. Yep.
But I thought the lab was just amazing. It was such a great place to be, and you could go
Mm.
to anyone. And we did things outside the lab too, which was
Mm.
cool to cement this kind of camaraderie. And
Yeah.
we became a big family. And when he had his 80th birthday 7 years ago, we
Mhm.
all went to Caltech, and I have this picture of us huge. It's like it fills a football bleacher of all of the what we called ourselves Baltimoreans.
There you go.
And it's all of us standing there, all the generations. And
Hmm. Mm.
so it was amazing. And I learned to think of science in a different way. Just do the simplest experiment possible, right?
Mhm.
And then if it doesn't work, you could figure out what else to do. I learned that from him for sure. And
Mm.
just think really high. Don't limit yourself. You can do anything, basically.
Yeah.
Well,
Wow.
here's all the resources. You have plenty of resources, right? You can do
Yep.
anything. And I took that to my lab. I said, whenever
Mm.
someone said, "I need to buy something for this," I said, "Yeah, just buy it. We don't have to worry about money. Because if you have an idea, just do it. I'm not going to limit
Yep.
you." Yeah.
Yeah. I love that. Yeah. Amazing. A unique legacy that will last, continue to last for
Well, the thing is, he trained
decades to come, right?
many,
Yeah.
many people who went on to train many, many people. So there's a huge legacy there. And I have done some science, but I also think the communication, which we've done since 2008, is also an outshoot of him in a way because he was always talking to the press. And like
Mm-hmm.
on a weekly basis, he would have people call him, "What's new?" So,
Well, yeah, and at a time I would say in 2008 and or early 2000s where the company line wasn't so much like it was sort of be skeptical of going on the record with the press, staying
so
in
it
the ivory
was
tower, right? And so that was that's
1979. I started my postdoc with him. I don't think
hmm.
you were born yet, Nels, right?
I was I
You
was
were?
in the mix. I was in single digits, but I
Oh boy.
but yep. Yep. Um,
Steve is in Hawaii. Really love TWiEVO.
oh,
If I had a do-over, I would have pursued evolutionary genetics.
yeah, good
That's
idea.
cool.
Yeah,
Cool.
thanks, Steve. Good. Thanks for being here. Okay, well, why don't we press forward? We came across, Vincent, you came across a really exciting, I think, game-changing paper, maybe sort of in the spirit of Baltimore big picture paradigm shifting science here. So let's jump in here. So the paper's title is "One Mother for Two Species via Obligate Cross-Species Cloning in Ants." Vincent, we're obsessed with ants. We can't stop with the
Yes,
ants.
we've done a lot of ant papers. Yeah, and I think it's a great experimental system, right? You can
Oh,
study them there's so many of them, right? They're like
well,
viruses.
there you go. And that's, I think that's absolutely bubbling under the surface here is because the populations are so big, the diversity so vast that they've done all of these natural experiments. And then as we, as humans or as scientists, just look at them or look at their genomes, all of this new biology sort of falls right into your lap.
Yeah.
And that's the case here. So, usually, when we kind of get to a topic that we've been on before, I'll go back and call out previous TWiEVOs, but we've covered the ants so many times, so much recently that I think we'll sidestep that for now. and just sort of concede to the ants as an incredible system to understand biodiversity, to understand the evolution around us. And from the, complicated title, but already, hopefully this sort of hints at something that's pretty weird. One mother for two species. What's going on here? And that's sort of like a, shot to the idea of what's the definition of a species. How could one mother give birth to two distinct species? So of course we're now, have to think about the title for TWiEVO 118.
Oh, at the end, Nels, let's show all the titles
Okay.
that
Yeah. We're
came
in
up,
the,
because this paper inspired so many titles, because the writing is really remarkable. You'll see.
Oh, the writing is, this is a paper worth picking up. Open access, published last week in Nature. And let's just shout out to the authors here. So I think there's some co-first authors. Most of these groups are, or all these groups are in Europe. So in France and Switzerland, in Italy, Spain, Austria, and senior author coming out of Montpellier in France, University of Montpellier, is Jonathan Ruminger. I'm sure I'm mispronouncing that. Apologies. Some co-first authors here, I think 3 co-first authors. And that kind of makes sense given the nature of the work here. So it's combining field studies, field biology, observations of ants in the wild, which in the geography here will matter, combining population genetics, population biology, some phylogenetics that we'll dive into, spend some time with. And then finally, lab experiments, bringing these colonies of ants, these are harvester ants, we'll talk a little bit about that, into the laboratory to sort of test if something as wild as a mother giving birth to two species could be a thing, or what does that mean, how to unpack that a little bit. So huge splash in the field of evolution, and an attack on that sort of traditional view of a species, and sort of the notion that two highly related, that there are barriers, that only highly related individuals will be able to produce fertile offspring. And so, obviously a massive theme for evolution. How do you sort of pass along genes from generation to generation, kind of into time immemorial? And all kinds of interesting cheating and conflicts and tricks that show up when we think about the definition of species, or why things reproduce sexually.
There's big time cheating here, man.
Yeah, so we'll be talking about sperm parasitism. We'll be talking about
polyandrous, having more than one husband.
Correct. Yeah, all kinds of sort of social gymnastics here for the eusocial insects to form the complicated colony, or for selection to act on all of these sort of exotic mechanisms and how they work. So, so hybrids are hybrid species so it's very common for species that are not closely related to hybridize, to share their DNA. And in fact, when we think about hybrids, think about donkeys mated with horses to make mules. Very familiar sort of concept. But there's
which humans have done. They don't do that naturally, right?
No, correct. Yeah. But so all these sort of possibilities, and with sort of infertile offspring that come from that. The ants have been doing this naturally for even longer, obviously, than we have. There's also, as these chromosomes are exchanged between two parents, all kinds of room for cheaters to emerge, things like segregation disorders where chromosomes outcompete other chromosomes. That can lead to, if the cheating chromosome is a sex chromosome, either an X or a Y chromosome in sort of the diploid mammalian setting, that can lead to sex ratio skews. You can go up to, populations might go to 99% male. There's no females around. The whole population crashes. But because of that selfish chromosome had an advantage to get into the next generation, these sort of out of control sort of scenarios can show up and break the rules of Mendelian genetics. The laws of genetics sort of go out the window, at least for these short periods. And then and we've talked about this, Vincent, on TWiEVO, like, why do species have sex? You're giving up half of your genes, half of your genetic material to a partner. And if you're really trying to maximize genes going from generation to generation, that's sort of like a short-term compromise that seems almost like, why would you do that? And in fact, there's a lot of populations that do just clone themselves or undergo asexual reproduction. But the ideas that have sort of built around why these sort of different life strategies is that those are sort of fragile or very dangerous. It's dangerous to go asexual, to just clone yourself, because genetic diversity collapses down to being identical. And then these populations become vulnerable to things like viruses. So we can't, the viruses are everywhere. And they're sort of pulling the levers behind the scenes here, even for these, behaviors by populations of animals or other critters. And so, yeah, so reproduction is a hot zone of evolutionary complexity, and that's sort of where
Yeah.
we pick up the chase today with these ants, and a really interesting puzzle of ant biology. So that was identified in this specific species of ant. So what is the species? It's the
Well,
Iberian.
before
Oh,
you go,
please, yeah,
I just
yeah,
wanted
yeah, yeah,
to
yeah,
mention some general things that they talk
yep,
about in the first
yeah, good idea.
paragraph. So they talk about how clonality is straightforward, right? You just
Mhm.
take an organism and you duplicate it. But
Mhm.
they say most animal species take a more complex route, which you were just talking about. So in
Yeah.
sex, you need. This is a funny way they write that. It requires the interaction of males and females. I
Yeah,
just
not
thought that
always.
I thought that was funny, right? Not always.
Yeah,
That's
yeah,
right.
yeah, yeah, yeah. Well, it depends again on your view. So you can back out. I'm fond of this because of my PhD work on a ciliate, a pond critter that actually has 7 mating types. And so in that case,
Hmm.
depending on the temperature that the cells find themselves in, they'll switch mating types in ways that sort of add even more diversity or more
Yeah.
possibilities of these things coming together. And so, yeah, just, I mean, there's countless stories of ways that biological complexity has emerged around the notion of reproduction. And it makes sense, right? There's strong selective pressure on what
Right.
genes
Right.
make it to the, and so you do all of the tinkering through mutation and sort of diversity, and then the thing that works, even if that's short term, might sort of pop
Yeah.
up and then turn over, and you just keep doing experiment after experiment over long distances of evolutionary time.
They also point out that it's complicated because you have to have two different morphs, right, of the same species, males and females. And they say sometimes there's even multiple females, depending on the season and the population
Mhm.
density or even social castes. So
That's right.
it's it gets complicated. But they say there's always one constant is that it's always the same species as far as we know, right?
Uh-huh.
And now this paper comes along and
Oh,
says,
yeah. So if, in terms of a mother giving birth to the
yes,
offspring of the same species, exactly. So underneath the species concept, I think, is this notion of you might hybridize with individual from another species, but then how you sustain that as a reproductive strategy is not clear. I think where the ants come into the picture here really beautifully is that they already, sort of the life history or the biological organization of the colony. So the
yeah,
division of labor, the workers, et cetera. We've talked about this very recently in some of the wild sort of ant um, adaptations or flourishes of evolution here. This one really, I think, plays off of that sort of colony structure. And then how does genetics sort of experiment on top of that? And that sort of will come out here. But let's introduce the players here, or the species at hand. So it's the Iberian harvester ants, Messer ibericus. The harvester ants are seed collectors, Vincent. They go around, they pick up seeds. They use their mandibles not to amputate the legs of their other guys, but to amputate the seeds a little bit so that the seeds won't sprout. They bring them into the colony and have kilograms of these seeds that they sort of collect to make it through the winter. So they have all this nutrition. So, to pull that off, they've figured out these division of labors. You have the queen ant, very familiar, and then you have the workers, et cetera, et cetera. And so one of the kind of the puzzle that they start the story with is that as so obviously the just the ecology is fascinating here and has a big impact on habitats and ecosystems, interactions between other species as they're messing with seeds and collecting them and harvesting them. But as you look at the genetics under the hood of this species, including the workers, what was sort of a real puzzler is that it looks like a hybrid species,
mm,
meaning it's got this genetic variation. It's got, one chromosome looks like has, the set of nucleic acids, the letters, and the other chromosome looks very different, to the tune of something like 43,000 genetic changes in these hybrids. And so, fair enough, that might be the simple explanation might be, okay, you've got that horse, you've got that donkey and they're making mules. And in fact, that's like pretty clear. And there's other cases of this for other ant species. But what that means then is you have two species that are overlapping in their host ranges. And so here's where the field biology matters, right? Because the puzzle here is, hold on, these guys are making these hybrids, but I don't see that other species around anywhere. The range, so, now you can sort of, and they do this, they compare 5 close relatives, modern relatives of Mesor ibericus, the Iberian harvester ant. And what they see is that there's like kind of the diversity you'd expect with common ancestor tracing back something like 5 million years ago. That's, by the way, that's about the genetic distance between us and chimpanzees, just to put it in a little bit of perspective. But what they notice is in those hybrids, one of the other species, so this is Mesor structor, and I don't think it has it's just another harvester ant. I don't know if it has a common name. It looks like there's a lot of that genetic material showing up in these Iberian harvester ant genomes. And yet those colonies are about 500 miles away from any known colonies of structor. And so the horse and the donkey aren't together in a way that could make the mule. And yet these colonies are teeming with mules, so to speak.
So the difference is that the structor workers that are produced, which we will see, are produced by the Ibericus queens. They can also have offspring.
Absolutely.
And whereas the mules are sterile, right?
Correct. And so we need to talk a little bit about the ant biology here. So and I think this is where actually sperm parasitism will come into the conversation. So let's and actually we can turn back the clock here, kind of retelling the story. But at some point, and including today, there are overlapping ranges of these two different ant species diverged something like 5 million years ago, but they still live in some cases in the same places. And so they have access to each other. And so the queens of the harvester ants, the Iberian harvester ants, are known to, mate with males from fertile males from both species and to collect that sperm. And so that behavior has been seen in many ant species called sperm parasitism, which is the female, the queen is in control here, is choosing in a sense biologically between the sperm that it uses to make offspring. And so what's useful here, it's a symbiotic relationship. The queen uses the sperm of the other species to make hybrids, males that are infertile, and those are the workers. And so that, again, it's sort of that genetics is then written on the behavior of the colony. You need all these workers out there collecting seeds for the winter. And so this is just it looks like selection is acted in a way that the queens have an advantage by using that sperm from another species to make a lot of these workers. And by the way, it's 2 for 1, right? Not only do you have sort of the production of all these infertile workers, but they're immediately hybrids. They have that genetic diversity. So these are all clones of each other, but they're clones that have all of that variation packed in, at least with that other species. So
but if I understand, the queen cannot mate with her own workers. That she doesn't, right? She doesn't make offspring. She only can do it with the other workers.
She can. So she can still mate with those ones, but there's an advantage to mating with these ones. So think about it this way. If she were only mating with her own species fertile males, and those show up from time to time, rarely, we'll probably have to dial into figure 3 here to see, like, it's all kind of laid out who's mating with who. It's pretty complicated in these ant colonies. But so if she mates with fertile males of her own species, and those produce clones, those clones are like, they don't have that diversity. They don't have that. That's a pretty fragile strategy. And so it could be that there is just sort of this like inbreeding depression by doing that. Whereas if she uses the sperm from these other males, immediately she has the hybrid vigor sort of in one step. And that could be a more robust genetic strategy.
So now let's look at the paper. Okay. The first page, that first subhead, queens depend on another species' sperm. The first sentence is, M. ibericus queens are unable to produce workers without mating with males of another species.
Right, to get those workers, she can still mate to get these. I think she can still mate with the other males of her own species, but not to get workers. That's absolutely true. Yeah.
And the work, as we're going to see, that her colony consists of Ibericus structure workers and some Ibericus workers as well.
Exactly. Right? And so the implication is there's an advantage from having both around. And in fact, now she can't choose. She needs that sperm to expand the clonal the workforce of the colony. She's become addicted to it in a sense.
But when you have a geographically distant Ibericus instructor, her mating happened a long time ago, and her colony still persists with that genetic information from the other species, right?
No, and so this is where it gets wild, right? So she needs, exactly. So she now needs that, and so so because she requires that mating with the males to make those workers, then what you could say is, oh well, she's stuck. The colony will always have to be co-located with that other species if they're going to make workers, and so that's where the genetic sort of trick happened, where if evolution, tinkering, trial and error allows you now to domesticate that male of that other species so that you're producing them, and now we're getting to a mother producing offspring of two species, then you can just take the whole kind of crew with you, let go of the original, the structure sort of co-colony or the overlap for mating opportunities, because now you've domesticated that other species to be part of your
That's the other word that I liked from this paper, domesticating males. Right?
Absolutely. And I think they've got good evidence for that. So we'll get to the implications here in a moment, but let's sort of walk through how they came to this sort of wild conclusion that the mother has domesticated males in a way that she still has offspring of males of her own species,
right?
But offspring of males of this other species. And then is it still that other species will become a question. Okay, so combination here, fieldwork, population genetics, lab experiments. And I would add to the mix here is patience. This study, took more than a decade. And in part because I think the finding is so wild. It's kind of, a new observation for what's possible in terms of that species concept. And so they really had to take their time to kind of dot the I's and cross the T's. It's also not, I think, super simple to just like bring the harvester ants into the lab, recreate a kind of colony structure, and then to have just the lifestyles here. They make a ton of these infertile clones, the hybrids, generation 1 hybrids, but very few fertile males of either Ibericus or Structor genetic backgrounds. And so just getting your numbers high enough that you believe with your eyes what you're seeing, which sort of breaks some of the rules of genetics. Okay, so punchline, the queens need the sperm of the Structor species to make these hybrid worker males that are sterile, but in the twist, they also produce these fertile Structor males. And then that takes that whole other colony out of the mix, and in that sense, they've domesticated them. Okay. So I think we talked about sperm parasitism, and that's known for a number of species of ants, but this goes beyond that. So how did they sort of get to that next level of the biology here? So they sequenced the genomes of 390 individuals and asked about genetic relatedness. This is among 5 different species of harvester ants that are sort of co-occurring here. And what really stands out is that the genomes, the hybrid, you see these hybrids exclusive to Ibericus. And the reason that's strange is that if you put this on a map, so you can, and that genetic variation, as we've mentioned, is for the Structor species, not for, Ponticus, McCarthyi, or Muticus are the other species of harvester ants here. It doesn't fit into that category. So then they take all of the distribution of M. Structor, the distribution of M. Ibericus, put that onto a map, and you can see in some places they overlap. I think it's like, southern Italy or eastern Spain on the coastline, the Mediterranean Sea. There it is. But in other places they don't overlap. And so in particular, if we go south to the island of Sicily, so now you've got this colony. Where's my geography? Or
that's Sicily here.
Okay, yeah. So, right, it's only blue. There's no red. So the distribution is only there. You're on an island, and you're about 500 miles away from any of the structures. And yet, if we look on that genetic tree to the left there, all of the hybrid workers have all of the, half their genomes basically appear to be Structor. I would note, so since we have this up, look at M. ponticus. There's one worker, so their sample size is a lot smaller here. I think, they spent a lot of time getting out in the field collecting individuals. They have something like 200 plus Ibericus. They only have a dozen ponticus, the closest relative. But one of those workers, even just of the 12, is hybrid. It has a lot of SNP heterozygosity, meaning it has a lot of genetic variation. If you just compare nucleotides, there's a lot of differences there. I'm curious who that maps onto. And if you collected 200, would that sort of pile add up? And is ponticus sort of experimenting with some of the same genetics to their mothers? Maybe are they involved in the sperm parasitism that could lead to a mother having offspring of two different species? Possible. And it could be at a different evolutionary stage where it hasn't been sort of advanced as far as what you see in the case of Ibericus, where all of these hybrid workers like that they collect, right, have that sort of quality. For ponticus, you can see there's the dot on the front. There's a worker that's totally, its genetics are ponticus related. And so interesting side point there. They don't really pick up on that. Okay. So not only, Vincent, do you have the different genetics here, which takes a little bit of work. You got to sequence genomes or do SNP mapping, et cetera. But you just look at them and they look different. So if you just grab the males that are coming out of the, that are being born from the Ibericus queens, the fertile males are hairy, and which looks a lot like structure, or sorry, the other way around. So the fertile males are hairy, which looks like Ibericus, and the hybrid males have less hair. They look, instructor looks like that. And so they have the appearance of this other species. So the female is having, is giving birth to rare fertile males of both species, and they look different just by looking at them. And that correlates with the genetic differences, that they're hybrid, they're hairless. If they're hairy, they just look like Ibericus. Okay, and here's where another sort of foundational part of genetics really plays to the advantage of the authors, is that they look at in addition to the nuclear genomes, which are hybrid, they can look at the mitochondrial genomes and ask, well, what does that look like? Because remember from our high school biology classes, mitochondria are passed from mother to offspring exclusively. Males never provide the mitochondria for their kids. That's provided in the eggs during when the sperm meets the egg. All the mitochondria is from the egg. And so when you look at these hybrids, right, that have all of the DNA of structure, or the males that appear that are 100% structure, which we'll get to in a moment, they all have the mitochondria of Ibericus. So what that says is that there aren't sort of males sneaking in from the structure colonies that got their mitochondria from a structure queen. All of the males that you see have the mitochondria of the Ibericus queen. Right. And so that's a pretty good sign that that it's really true, or a good piece of evidence that this female is giving birth to two different species of ants. Okay, they're still not convinced. So next they go from field to the lab. So they describe that they have 5 colonies of Ibericus colonies that they've set up in the lab. And they just start collecting males and they genotype them. And it's about 10% of the males that they recover only have structure DNA. They're still not convinced. So then they spend a year and a half, Vincent, just watching a single queen having kids, giving birth to ants. And here's where, this is pretty rare, I think, maybe especially in the laboratory, to just have the kind of biological throughput to get your sample size large enough to kind of see patterns. So after a year and a half, they got 7 eggs that turned into fertile males. They're having, there's a lot of males being born, but most of them are clones, right? They're these infertile hybrids that become the workers. And so of those 7 eggs that actually turned into fertile males, 2 of them were hairless and 5 were hairy. And when they sequence those genomes, that correlation holds. The hairless ones have structure DNA, and the hairy ones have the Ibericus. So that's kind of it, actually. So that's sort of the, you'd say, like, kind of conclusive evidence, seeing it with your own eyes and connecting that this queen is having kids of 2 species. So how did this evolve, or how do you put together the history of that? And this is where I think that, this is also a good one, not just for kind of this week in genetics or this week in wild sexual reproduction, but this week in evolution. Using the comparisons, the phylogenetic comparisons, the relatedness between these, they can put together a story of how this evolved sort of over the eons, or however many million years, or million years that it's been. So they can just build off of the idea of sperm parasitism. And in fact, you can go to the queens and see the different sperm types from different species, um, and start to see how, this kind of adds that source of variation. This is sort of our idea that evolution walks down these sort of dark alleys and gets in trouble. And then you just sort of add this complexity on top of complexity. They become addicted to the sperm from this other species. And in fact, they find evidence. So what that would first imply is that then there are quote unquote, wild type structure males that are interacting with these queens, that are providing that sperm that then the queen depends on to have those. And in that overlapping part of the map, I think this is in like a little bit off the coast in southeastern Spain and northernmost Italy, they can see they can observe this in the field, that there in fact are wild type males there. And so that sort of gives you that first step in this evolutionary process, where the wild type males are providing the sperm to the Abericus females. But then that kind of gets them to that sort of addicted state, where then you can imagine the next step in evolution is to let go. If they're able to domesticate the males, to give birth to them directly, as opposed to waiting for those wild type males to just show up at the colony or go between colonies, then they can let go. They can expand their host range. And then you can imagine evolution acting on that, right? If there's just one queen that figured it out, and by the way, we don't know the mechanism, that's for sort of future work, which I think is fascinating. What does the cytogenetics look like here? What are the karyotypes? How are the chromosomes arranged? How are the genomes kicked out? Or what's the genomic gymnastics to sort of pull this off from at fertilization and beyond? Because you end up letting go of, or kicking out, the DNA of Ibericus to give birth to these fertile males that then bring or that you domesticated.
And we don't know that. Their DNA, it's all their DNA. There's no queen DNA in it.
No,
that's
correct.
The thing. It's really cloning, right?
Well, and that's the genetic, exactly, or eliminating the maternal genetic contribution.
Except for the mitochondria, right?
Except for the mitochondria, exactly. And so, the notion is that happened by accident, that you could describe that as a complicated mutation, but then selection acted because it was such an advantage, because now these colonies, right, have let go of the need. It's sort of like solving your addiction by figuring out how to make the drug for yourself, kind of thing. Like, you no longer need the drug dealer anymore. And so and then is there some advantage then, as they've sort of spread across the Mediterranean area in Europe, that's sort of implied in the work, but that would definitely give you a sort of evolutionary pathway for this to work. So figure 3, kind of wild biology here. Figure 3 is a good way of sort of trying to capture these evolutionary steps, what they call ancestral and derived. And so, so here we can see, if we start, there's a lot going on here. It's sort of a complicated family tree that we're looking at here, a pedigree. And so the ancestral state in blue, we have the first species, Abericus, and right at the middle, kind of top middle, is the female. And there's a line there to the red, to the structure colony. This is when they're sort of occurring with each other. And so in the ancestral case, the males, as usual, are mating with the females and giving birth to new queens, new females, as well as fertile males. Mm-hmm. And then you see at the sperm parasitism, that's where these hybrid clones come online and just allow you to have that cast of workers, these hybrids. And so that's where that first step is. And there's evidence for this. So you can see that kind of diagonal dark red line going to the wild type. So that are those observations that in co-occurring sort of geography, that sperm parasitism is happening with wild type or true structure males. Now as we march down from ancestral to derived, these are the ones that they find today that are not co-occurring. The ones on Sicily, for example, where there's no structure colonies nearby. So all of the reproductive biology is still happening on the Ibericus side, but here's where the cross-species cloning is happening. So in addition to making those hybrids, if you go in that diagonal to cross-species cloning, you can see now the fertile males that are structure-like have the chromosomes. This is still somewhat implied until you do the karyotypes, but they just have the genetic material of structure. And these are fertile. So now you've got these fertile males that the female. So I think the the way they wrap it up the end here is they say, let's see, what's the sentence? It's evidence that that the queens lay eggs of two species, but they mate with three, which is the domesticated structure, the wild type structure, which was also in the ancestral state, and then, of course, the males of their own species. And that's all laid out here in figure 3, which I think is a nice summary of how this is going. But those clonal males, so then that raises all kinds of cool questions about, those clonal males in the derived state, which have all the genetic material of structure.
Right.
But do you see, so what would happen if you take those males and if they find themselves back in a structure colony? So this is like the domesticated species returns to the wild. And so they address that a little bit. So in their field work, they don't see that. And so that implies that as it's been domesticated, it's let go of the ability, right? And that's pretty common to a lot of domestication genetics, I would say. But what they haven't done, or at least haven't reported, and again, this isn't like criticism, this isn't podcast peer review in the sense like that the paper loses any of its impact or sort of excitement. But, the experiment you'd love to do is to take those domesticated males, purposefully or experimentally mate them with the queens of the structure, and then see if any offspring come, or has it sort of let go? Has it lost that ability to sort of return to the wild? Yeah. Really
so they looked at 45 structured genomes to see if they could find hybrids, which would be the consequence of what you're saying, of the clonal males mating with wild type females of their own species. That's the key.
Correct.
And they don't see that happening. I don't think they did the mating themselves, right? They just looked at 45 genomes. But the other question is whether these clonal males can mate with Ibericus females, and the answer is yes, they do.
Yes,
right?
They do. Exactly. Yep.
And so, that's like you said, that's so. What's the mechanism there? Is it the mitochondrial DNA that's doing it? Who knows, right?
Could have a role in it, yeah. So to me, this sort of hints at a little bit of the, some of the cheating that can happen when we think about segregation disorders. So in that case, like there's not at the whole genome level, at the chromosomal level, you can outcompete the other chromosomes, or are they somehow outcompeting the whole genome? And if so, what's the mechanism? That's sort of going from the wild observation of a mother giving birth to two sons of two different species, all the way down to the molecular basis of it, which is certainly, in play. And I think the clues would come from, starting maybe with just looking at it, the traditional cytogenetics, taking karyotypes, seeing, but then also trying to catch the eggs right after fertilization and to try to watch that process play out. How do you go from having the genetic material from the mother, like what step of meiosis as the two gametes come together, does this sort of unfold in a way that only makes the offspring, where you lose your own species sort of offspring, which then solves or sort of feeds that addiction, because now you have all these fertile males of the other species that provide you the sperm that you need to make the massive set of hybrid clones that become the workers collecting the seeds.
Amazing, right?
Crazy town. So a couple of take-home messages here that I think are fun to sort of just step back and take stock of. So, I think first of all, just how bringing together the field work with the population genetics allows you to gain this view sort of these snapshots of an evolutionary process kind of allows you to turn back the clock or see things that you can sort of infer from gathering or putting together these two fields that have, historically been sort of, in a lot of cases, not, especially the molecular and the field work have been sort of separate. The genetics and the field work have come together a while ago. I think it also, for me, like it's in a couple of ways sort of shifts your thinking about what's biologically possible or genetically possible by sort of breaking the rule or having a clear example of like stretching into something that we probably wouldn't have predicted could be possible. So, and what do I mean by that? So two ways I'll put forward, and by the way, the authors do, and there's some perspective pieces that sort of hint at this as well. But so, we talk a lot, Vincent, about horizontal gene transfer, right? So mostly genes are passed from mother to offspring,
right?
But there are some rule breakers there, usually just at the one gene level. And viruses are often involved. Some of the work in my lab are involved in that process of shuttling a gene from one genome of a host potentially to another. And retrotransposons that encode that reverse transcriptase that David Baltimore discovered back in the 70s, part of RNA tumor viruses as well, seems to be part of that sort of gene by gene horizontal transfer. This is horizontal transfer, but done at an entire species genome level, right? Yeah, because you have this diverged species from so the last common ancestors is inferred between Ibericus and Structures 5 million years ago. And yet today you can take all that genetic material, bring it to the egg of the queen mother of Ibericus and transfer that whole genome and delete the maternal genome. The mitochondrial DNA goes on, but that's horizontal gene transfer because it's no longer, in one sense, if you kind of step back, because it's no longer the two parents giving their DNA. This is the mother sort of steps aside, and in this case, allows or the result is that you get the entire genome shows up horizontally. Also, I think another way maybe of kind of sort of conceptualizing the paradigm shift here is thinking about the scales of biology. So, and as cell biologists, as molecular biologists, seeing some of the same events or behaviors that have the same sort of conceptual style of what you see in the ant colony. So now the ant colony becomes the cell or becomes the biological unit. And the authors, I think, say that you can think of Structure actually like an organelle, that it's bringing in a new genetic contribution that sort of allows more interesting biology to happen. In this case, it's part of the colony is still going forward with Ibericus males mating with Ibericus females, but then now you have this sort of new way of adding these functions in that you become addicted to. And that at the cellular level, that feels very much like the origin of eukaryotic cells, where you get the mitochondria and it becomes an organelle that then allows for all of this new biology to happen in the same cytoplasm. Mm-hmm. Here, the different species comes in and allows for all of this new biology to happen in the boundary of the whole colony. The colony is like a cell, sort of in that analogy of the scales of biology here.
They invented a name for these females, right? Xenoparous, meaning they need to produce individuals of another species as part of their life cycle. Yeah. So xeno is different, and parity means to give birth, right?
Yeah, for like a foreign birth.
Yeah.
Xenoparous. I don't know how to exactly pronounce that, but yeah. And that kind of again underlines that we've never seen that before, and so they have to come up with a name for it. They've discovered something new, just happening right under our noses as these ants are just walking around collecting seeds. The deeper you go, the more fascinating it gets from a genetic standpoint.
So they also say that so as this pattern evolved, it was not good. It was a liability for the queen to depend on another species, right? Because they have to be right next to it. Exactly. And so they got the ability to clone them in their own colony, and that gets over that, which is a big step. It's not easy to do that, as we've been talking about, and we don't know how they were able to clone them, but obviously that was a big step in evolution that they could then move away.
Yeah.
So then I like this discussion about, so while trapped in a life cycle of a species exploiting their sperm, clonal males, the ones that are in the Ibericus colony, propagate their genome through the reproductive efforts and parental care of M. ibericus. In this sense, clonal males can be viewed as a perfected form of male parasites. Oh my gosh, right?
Yeah,
Because they're living in another colony and they're taking advantage of the
exactly.
resources of the colony, right? But
Exactly right.
they're obviously giving something also. So I'm not sure that's a parasitic relationship, right?
Yeah, so I think you'd call it symbiosis, but
Yeah. Yeah.
symbiosis doesn't is like the notion that everyone's so then we're getting a little philosophical, but the notion that everyone's getting along versus everyone's just addicted to like you just depend on this now. It's the only thing. There's kind of the same thing. It's just a sort of a different way
Yeah.
of
Yeah.
describing it. But yeah, so like in that sense, right? The males, the clonal males do all or don't have to do all this work. The queen of a different species does all this work, and so that sort of has that
Yeah.
parasitism to it. What the trade-offs are. So I think we hinted at this already a little bit, which is that by bringing in a hybrid, by domesticating it, you can sort of advance these asexual clones, but with the twist that you now have all this diversity or variation from the other species. So that could be sort of a compromise
Yeah.
along like somewhere in between complete asexual reproduction, which is thought to be the pretty brittle. So you're like, let's say your immune systems are all have less diversity because it's all of the same genetics, and at some point, very quickly, you can imagine an aspiring virus population that's mutating and getting around all of these immunological blocks will find a way, will mutate, and selection will act because these viruses will replicate as they defeat the immune defenses. Then that asexual style, lifestyle will go into a collapse because
Right.
there's no genetic variation where it just happens that you're resistant to that new sort of mutated virus, and so then a lot of collapse happens. But there's like that rescue where that new sort of immunological variant then becomes the founder of the next
Yeah.
set of that population. And so maybe this is like somewhere in between, right? Because you have this hybrid, but you're not really you're not recombining, because you're cloning, you're bringing in the two chromosomes and you're maintaining some level of variation, but it's sort of like a binary instead of an exponential sort of swapping and changing around, genetic material. And so I guess the prediction would be that you'd be sort of in this intermediate state where you might not be super stable in the sense that you can probably sort of this is not the right way of saying it, but evolve your way around all of these kind of pathogenic attacks. But you're not so fragile because you actually do have the variation that's sort of amassed over 5 million years, but it's binary instead of sort of exponential versus being just a zero or a one. At least having a 0 and a 1 is better than just having a 1 or just having a 0 would be the way I'm trying to think about it here.
So a couple of thoughts here.
Yeah.
First of all, they say something interesting, they say females control the terms of the relationship. I thought that was a really interesting statement.
Yeah, it's good.
So sex arose because cloning makes you more susceptible to parasites like viruses and other things. So I wonder if these clonal males are more susceptible, or does the unique structure of an ant colony protect them from viruses, for example?
Yeah, good question. So I think there's somewhere like the idea would be there's somewhere in between, but is there and I think that's an important point because all of this genetics is really being written on the life history of the colony and the complexity of it.
Yeah.
And as we've discussed, right, these ants aren't messing around. If an ant shows up back at the colony with a wound on its leg, there are species of ants that will amputate those wounds, right? Or there's plenty of workers, or I don't know I'm just not an ant biologist, but there are, ants that will, if they think that ants are coming back that are infected, they'll get rid of them. They'll kick them out of the colony, right? And so it could be that sort of this exotic sexual reproduction lifestyle of giving birth to two different species, in a sense, could only persist, or is more likely to persist, given all of the social engineering that's going on in that colony to keep infections out of there. It might buy them some time or some space where you can kind of pull this off. And so then as scientists just show up and are kind of confused, why are these ants that are 500 miles away showing up here? Yeah, I think it's a great question. Yeah.
Also, if this is a good strategy, is there more of it out there? We just haven't found it yet, right?
I think so. I think that Ponticus data point is sort of, I mean, it could just
Yeah,
be a
that's
single
true.
outlier,
Yeah.
but I think that's hinting at this. I think there's, this is probably the tip of the iceberg, honestly, is how I'd think about it, is that there's
Yeah.
all kinds
Yeah.
of variations on this theme or different kind of complexities, all these different blind alleys that these insects can walk down, and they've got the numbers. They've done all of the natural experiments. I would say, the other thing we've been talking about this as a mother giving birth to two species, but I think we have to be careful, or really sort of step back and think about how we define a species. So is it that this is now actually a mother giving birth because it's domesticated? And so I guess that's really where that experiment that we want to see, where it sort of really hinges on that idea that the domesticated could go back and mate with its own, original species. If that's true, I think that bolsters the idea that the female is that the queen is giving birth to brothers of two different species versus, once it's domesticated, it's now sort of takes on a different quality in terms of how we might think about it as a different species, maybe a third species in a sense than the original one and the Ibericus one.
A couple of comments here from our listeners. Steve, thank you
Great.
for your support of science communication.
Yeah,
really appreciate that.
Mm-hmm.
Andrew writes, they remind me of mosses and how they have both diploid and haploid members of a species.
Yeah, so there's all kinds of cool examples of this in the plant world. So I think they point to, sperm parasitism, for example, you see in conifers. In addition, yeah, exactly. This diploid, haploid, all of the yeasts obviously are doing this too. All of the kind of genomic gymnastics that can happen when we go out there. The ciliates are my favorite, as I was mentioning before. The 7 mating types that show up in some of these species, it can really be something. It maybe sorry, I'm going to kind of surf off of that a little bit to say, I don't know, the more we look at the natural world, how big things are, how big biology is, the sort of possibilities, it kind of grows your imagination, versus the difference between taking, a different view, maybe a more sort of current political view that makes our world smaller. You can only have 2 genders, or there's only 2 sexes, or, that just feels like we're letting go. I'm not trying to draw a direct connection here, but what I would say is just approaches that make us think about things that sort of grow our imagination based on the observations we see, or open our minds about what's possible. Give me more of that. I mean, it's just
of course.
Yeah.
So it's not always binary. That's what you're saying.
Yeah.
It isn't black and white. Yeah. It's there, everything in between. Yes, I agree.
And when you open your mind to the possibilities of what's out there, life becomes more interesting, more rich, more exciting, more inspiring, more everything. And so that's kind of in contrast, I think, to some of the headwinds that we're feeling these days in our own neighborhoods.
So Bowden writes, is this paper just documenting the logical conclusion to Lynn Margulis's thoughts on symbiosis?
Ooh. Good. Yeah. Wow. We were really drawing here. So do you want to do a little background on Margulis's ideas, Vincent?
I'm not good enough to do that.
Okay. I don't know if I am either. I was trying to punt here. Yeah. So, I think, so Margulis had some interesting ideas about, so in addition to mitochondria being like one of these key symbiotic events, is the flagella. Also, like some of the other cellular structures that we see, how do these come together? I think it has, I would say, how I'd say it is, I don't know if it's a logical conclusion, but I think it is it feels like it's in that sort of really creative, having an open mind to things that might break the rules, or things that you don't expect.
I mean, she basically said that mitochondria and chloroplasts came from free living organisms way before we had good evidence for it, right? And then the ultimate symbiosis, people didn't believe it for many years, right?
Yeah. And then it went into some kind of in more adventurous space, I'd say, as well. And I don't think I can do that justice without sort of refreshing my memory. But yeah, no, I think there's definitely some lines to be drawn here. I like that comparison.
But here, let me go back to Bowden also says was this result not predictable?
If you predicted it, like my hat is off to you.
I
That
wouldn't
is,
have.
yeah, I wouldn't have. Um, I think hindsight's 2020, and if you're following um, the field of sperm parasitism, for example, and even I think cases where there's reciprocal sperm parasitism, so like the sperm of one species or their queens in these insects, I think it's Hemoptera is the order or something like that. That's not new, and so this like takes that first step, which in seeing some complexity, but it takes it to the next step, and I think does so convincingly. I guess that's where I would say that yeah, hindsight's 2020, but
Yeah,
in
of course.
someone who predicted it great, I wouldn't have. My hat's off to the authors for putting it together.
Troy wants to know if mitochondria have autonomic DNA.
Ooh, I don't know what that means. Do you, Vincent?
It has
Autonomic?
it has its own genome and it replicates independently of the nucleus, right? And it's expressed independent.
Oh
So I
yeah,
think
yeah, absolutely, yeah,
right,
yeah,
and it's maternally inherited, right?
correct.
Um, Pete,
That's another fascinating entire
yes,
field is so some of as that symbiosis has played out over billions of years, um, the genes that have been so again sort of horizontal or gene transfer from mitochondrial genome to nuclear genome, and what are the genes that make it, what are the ones that don't, that
mm-hmm.
remain in the mitochondria,
Yeah.
and so um, yeah, no, another fascinating, a lot of discovery is left to be made, a lot of active labs
So
thinking about it.
Pete says, don't we have a banana tree crisis because of single clones around the world and fungal disease?
Yeah, and so that's like the heartbreak of domestication, right? So you start to inbreed these, varieties of bananas. Your genetic or the load, I guess the genetic load, it's sometimes described. Um, the bird like builds. You have inbreeding depression. You have less diversity, and then if a aspiring fungal pathogen shows up or another pathogen shows up, those populations can be really vulnerable because they're stuck. They don't have there's not the variation. There aren't resistant, clones out there that can counter that. And so that's the central prediction of the red
Mm-hmm.
queen hypothesis, that the reason you have that you give up half of your gametes is the sort of gift of recombination, that by diversifying the genomes in the population, that then there is that sort of lottery winner that's resistant to that fungal disease that gets you through. And so it's sort of like, you've had this great banana clone running and you've built an industry on that, but it's sort of the trade-off is that it's become fragile. It's brittle to pathogens.
Milos, thank you for your support of science communication. Really
Appreciate it.
appreciate that. Tryon wants to know if there are viruses that replicate in mitochondria. Yeah, there are.
That's
They
right.
seem to be derived from so the mitochondria were once free living bacteria or alpha proteobacteria,
Right.
and they had their viruses, and so the viruses in mitochondria, mitoviruses,
Mm-hmm.
seem to be offspring of those original bacteria. Yes, so they are there.
Life finds a way. And the viruses are everywhere, pulling the
Life
lever.
finds a way. So Pete says, are these male structures now a single clone, not getting any sexual diversity from lack of females? Yes, that's right, right?
Yeah.
They're cloned. There's just straight clone. They get no genetic information from the females. And
Yeah.
how that happens is really interesting, right?
Exactly. And so that would be interesting too, right? Is to then, how many times has this happened, right? So if you go among the colonies of Ibericus that have these domesticated structure males, and I don't know if they did, or so are they all clones, like meaning so that happened once, the same, right? That in evolution, that they figured out how to domesticate these, and then those are the ones that spread out across, um, you know, Spain, etc.
Yeah.
Or has this happened multiple times? In which case, there would be clones that would be a different sort of snapshot of that domestication process, and then that would become fascinating to kind of plug and play or mix and match those. But my guess is that a single clone, and And so it's that compromise, right? So they do have as a when you take the worker cast, the hybrid males that are infertile, you have that sort of variation from the two, but you don't. But yeah, as the domesticated males, they could go the way of the bananas, and so in which case then you need to sort of recreate if you're down the alley, the dark alley, with needing the structure sperm, and so you'd predict that either you would need the structure males the wild type males to show up to expand their range, or your
Mm-hmm.
range would contract back. Yeah.
Tom reminds us that Lynn Margulis was sympathetic to HIV/AIDS
Yeah.
denialism, off the rails thinking. No excuse for that,
Yeah.
because
And I,
there's nothing. I mean, Nels is telling us to think out of the box, but when you have something, a virus causing a disease, there's no need to deny it, right?
well, right. And so there's kind of circling around the history of the Red Queen hypothesis is this faculty member, Lee Van Valen, who had all these kind of quotes. It's something like, it's good to have an open mind, but not so open that you'll let everything sort of fly in there and give it the same, sort of the same weight.
It's
And
good.
so, yeah, no, Margulis, I think, was a free thinker, but maybe kind of got off the rails in some important places, as like you're pointing out there, Tom.
I don't know how a smart person can do that, Nels, right?
Well, I think I'm going to say peer review is a good thing. Feedback from your friends is a really important thing, especially as, like in science, in our worlds, as you sort of advance. And if you, well, I think this applies to anything, any human condition or any human pursuit. As peer review goes away and you have success, the problem is you sort of embrace your own your own voice, or you trust you trust your own voice so much that you get the confidence to say things that might not actually be real, and to advocate for those things. And so having that, this is kind of why when you and I pick up a paper like this one, we bring to it that criticism, right? That sort of skepticism. Is this real? What's the evidence? How does that work? And there's a real danger, I think, for our species if we let go of that impulse to be critical or skeptical, not just for the things that we might sort of, take, quote unquote, doing our own research, things, information that we might take that doesn't turn out to be true or benefit us. But on the other side, the person who's purveying that, let's just go to RFK Jr. for a moment here. Some of the ideas he's putting out there, I think, could use some peer review. He could use some feedback from people who actually know how to weigh evidence and to put that into some context. And as soon as you let go of that, things can go off the rail. And especially if it's someone who's had success or is someone in a position of power, oh my God, you can go off the rails fast. And the impacts of that.
Sure. Lysenko, right?
Yeah, that's a perfect example. Yeah, Lysenko in the Soviet Union.
So in science, there are some rules, right, which we know don't get broken very often. We had this rule that only two same species members will make fertile offspring, and now this paper breaks it. So you have to be open to this stuff. But when a virus is known to cause a disease, does HIV cause AIDS? Yes, no question. Does it always? No, there are some people who are called elite controllers that don't get sick, yet they're infected. Yep. So it doesn't mean that it doesn't cause AIDS. It does in millions and millions of people, right? So it's always a nuance, and maybe it's a matter of overinterpreting the data, right? She saw some people who never got sick, and she said, okay, the virus doesn't cause AIDS, but that's not how it works in viruses.
Yeah, not how it works. I think the other layer here that's important too is, good faith, bad faith approaches. And so I think if you're approaching something in good faith, like air it out, but there's plenty of cases where it's very easy to move into bad faith. You're using this information or this influence in ways that do you really care? You don't care to get to a closer understanding of the truth. You care about using opportunity to increase your power or, command more, whatever it is. And so that's another, I think, piece of the puzzle here as well. Yeah.
So Vanity points out that a poet made up the term variety is the spice of life. Yeah, but of course that Can be used in many, many ways, right?
Absolutely.
And Les wants to know if these male ants are clones or just brothers.
Oh yeah. So the male, the Ibericus male versus the domesticated um, uh, structure males, they're brothers. They have the same mother, brother. So there's this there's this great phrase that the young people use, which I think is mostly just old people trying to act cool. Which is, you know, like for a close friend, you say, "Oh, you're just a brother from another mother," or a sister from another mister. Here, nature's reversing that. It's a unrelated brother from the same mother. So by the definition that you share the same mother, you're a brother, regardless of your genetics underneath that, right? The same queen gave birth to the two different species. And so it's a mother of an unrelated brother, is what I would say.
Anity says, maybe my response to the do your own research people will be to make sure you apply skepticism to what you want to hear as much as you apply it to what you don't. Yeah, I mean, that's a problem is when you Google and you find something you want to hear, you tend to believe it, even though it may not be right.
Exactly. And sources of sort of authority here. I mean, this came out a little bit, I would say, Vincent, in your conversation with David Baltimore that you reposted, is he was saying how, you know, the challenges of communicating science that we can, you know, if we say DNA, everyone kind of knows where we're coming from. But as soon as we say ribosome, we've lost, you know, 90% of our audience. And so that feels to me in that space of that you're bringing up, Vanity, of doing your own research, is how do we bring back that level, that sort of baseline level of knowledge, or even the foreign language of biology? If every fifth grader knew what a ribosome was, then I would feel a lot more confident with people doing their own research from that basis of knowing kind of what this biology is. One of the problems here, right, is even so, I think there's plenty of people applying skepticism, but it's a little bit out of context of how biology actually works. And so the skepticism then comes from, you know, when or plays out where RFK Jr. says, you know, don't trust the experts. That's authoritarianism. Trust me, the non-expert. While he's talking out sort of both sides of his mouth, saying, don't take my medical advice. Listen to this crank who doesn't know what they're talking about and is making a bad faith argument about taking a horse dewormer for your COVID infection, or using peach pits to treat measles, or whatever garbage he's purveying from day to day. And so I think the trick is in that space, which is for that person to exercise I think all humans are exercising skepticism, but to do that with a basis of knowledge of how biology actually works. And that's a really tough question, or problem, or obstacle or challenge, is how, especially as we look at the headwinds that education is feeling at all levels, and the investments that are going away, or the attacks on that, is really solid early education in science, in math, in the STEM fields and beyond, in the liberal arts. A topic that's under massive pressure here in Utah is the state legislature wants to have everyone do STEM as if somehow everyone's going to make more money. And it's really out of balance. And so I think the danger is if we, that person who's quote unquote doing their own research, it's very easy for us to sort of sit back and say like, oh no, you're out in the weeds. But there's not really by just saying that, I don't think is a pathway back to getting someone out of the weeds. It's starting at the beginning and trying to rebuild a shared language or a shared knowledge as a baseline here. I don't know how to get there.
But now, even though people may know DNA, that's all they know. They don't know anything about it. For example, if there's a little DNA in your vaccine, they think it's a bad thing, and they don't get it. They don't understand the nuance, right?
No, I hear you. And so that's where, that kind of gulf almost between the way they're talking about, think about, genetically modified organisms, GMOs, those will sidestep vaccines for a moment. And people saying, oh well, there's RNA in that genetically modified thing, like because they put so people put microRNAs, for example, I think into potatoes, so there's less acrylamide when you fry them. McDonald's is going to launch this potato that will have less acrylamide. It'll be actually, it's sort of trace amounts, but it'll be slightly more healthy. And there's a way to do that with the microRNA. And then people that got shot down because it's like, well, oh no, there's RNA now that in that, not realizing that RNA is in everything that, in all of our cells and everything we eat already. It's like, yeah. Um, and it's easy, like that's the challenge here though, right? Is it's easy to say, oh, that's nonsense biologically, but we're very lucky that we have that sort of, we have that, foundation that took us decades to build in terms of our
Yes,
knowledge
sure.
of genetics and molecular biology. And
Well, we have to
so,
share it then. As we have to share it in
absolutely.
a non-condescending way, which
Yep.
we try to do. And
Yeah.
but on the other hand, people have to listen and they have to not reject expertise,
Well,
right?
yeah.
Which
Right.
they
And so,
do. We have a president who says, "I like uneducated people," which is the most friggin' insulting thing to anybody. So
yeah.
uneducated people should never vote for him. But
Yeah.
this is where
Yeah.
we are. Even RFK has said,
Oh, yeah.
"Don't expertise is not how a democracy and how science works." Are you
Right.
shitting me? Of course, it's how science works. If you don't
He's
know what you're talking about, you don't get anywhere.
Yeah, of course. No, that's right. And so, but it's in that sort of skepticism which exists. I mean, let's like GMOs have for decades been up against it. Vaccines are now right in the, squarely in the spotlight. And so, but I think it's interesting to try to step back and think about like where does that skepticism come from? So, right when we get onto an airplane, um, or you get onto a train here in an hour or two, we kind of trust that the person driving it or the person flying the plane,
Of
that that
course,
that those decades of training, all of
yes,
those hours of experience, like we're pretty comfortable with that notion that that's who we want behind the controls.
yeah,
And
expert. He's
so,
the pilot's an expert, right?
and I would ask people right in this exact moment, right, of like that, well, why should I believe you talking about a GMO or whatever? In that exact moment, to try to like, how do we bring that kind of framework or perspective into it? Like if I just said, okay, here's your choice. You can get on this flight across the country with a pilot who's been doing this for 30 years, or I can just draw a marble out of the hat and we'll just pick some random person, myself included, sitting in the chairs here. And then asked, okay, we're doing that. Plane A is the pilot with 30 years. Plane B is me or someone else who just got picked out of the hat. How many people do you think would go on my plane? I wouldn't want to go on my plane. I want to go, I'm going with the pilot, right? Who has that experience. And yet here we are where we have people, heart transplant. We talked about this, right? People going in for heart transplant. I will trust the surgeon to literally take the heart out of my body and put in someone else's, but I will not trust your advice on a vaccine. It's just disconnected from reality. It just,
Well,
it's
part
wild.
of the narrative is that if you say something, RFK will say, "Well, you're just getting paid to say that," and this is just BS because
Yeah.
most people are not being paid, and he has nothing else to say. He has no other retort, so he
Yeah.
goes always to the same thing: "You're being paid," and people
Yeah.
should begin to realize this. He has nothing concrete to say against whatever it is that you are saying, except either you're lying, or you're
Yeah.
wrong, or you're being paid, and those are the signs of someone who knows nothing.
Yeah. But it is pretty effective, at least so far. Hopefully, not for years to come. Hopefully, just for a shorter amount of time to come. But yeah, that is pretty persuasive somehow for enough people, um, by making that argument that there's that good faith, the assumption of bad faith, versus good faith, incredible
I
disconnect
mean,
here.
this is a minority of people, I think, who feel this way, but
Yeah.
the election has been gamed in such a way that they have prevailed, unfortunately.
Well, and the power structure is all out of whack. I mean, the Republicans just can't stand up to, or are trying to figure out ways to stand up. I don't know if did you watch any of the hearing last week or whenever it
Oh
was?
yeah, sure, sure. Yeah,
Bill
yeah.
Cassidy got up and said, "Well, do you agree that Donald Trump should get the Nobel Prize for
Yeah, yeah,
Operation Warp Speed?" And RFK Jr., "Oh yeah, of course, of course." So then why are you against the COVID vaccine if it's so successful? And even Cassidy, right? He's the way he's framing his criticism of RFK is, oh, well, at least we agree that Trump deserves a Nobel Prize. Wild, just absolutely wild.
Well, also he's lying because he's just saying yes to please the man when
Oh yeah, of course. Yeah.
he doesn't agree because he doesn't think vaccines are of any benefit. But these are the contradictions that you can easily pull out of people like that who have no logic behind their conclusions.
Yep.
Unfortunately, I hope it ends sooner rather than later because
Yeah, you and me
people are
both.
going to die. I mean, the guy in Florida is not going to have kids get vaccinated to go to school. This is a stupid idea. This is just stupid and ignorant and inhumane. Okay, and
Yeah.
it's a ploy to get votes. And I did a live stream with Dan Wilson last week. He was offended at him using the slavery word to describe making people get vaccinated. It's just ridiculous. It's public health. It works, folks. It's not about disdain. How can it be about disdain? We're trying to save your life. Where is your brain? Right, Nels? Okay. Enough of this.
Yeah, we could keep going.
Well,
Okay.
the problem is everybody here is we're preaching to the crowd here, and the people who don't are not going to listen to us.
Well, right. So persuasion around this, and I think, well, kind of circling back to the beginning of this rant that we've gone on, would be that foundational knowledge, and how does that come into the picture?
I so admire experts in every field. When I hear a musician playing beautifully, that's an expert. I love it. When I see someone making something, a house or a piece of clay or a work of art, when I see you doing experiments, these are all experts. It's beautiful to see how a human can become an expert and do something so friggin' wonderfully. I watch people play guitar flawlessly and beautifully, and I'm like, oh, I can't even come close to that. And I admire it. I don't reject it. I mean, expertise is everywhere, and the people who reject it are fools, essentially. They're fools.
I like that art. I like the celebration of expertise.
Now, these people who say we don't like expertise, they're going to go to a concert and hear some expert being brilliantly musical, right? They're full of it. They're just full of it.
Well, yeah. Okay, let's move on. So let's close out here with we'll go into a slightly different topic here, our science picks of the week. And I've got one, I think, Vincent, that's kind of fun here. It's pretty inspirational, kind of stepping away from some of the headwinds and challenges we're talking about here. So this is a article that came out. It's a guy kind of in your neighborhood, so to speak, and he's on the faculty at NYU. He's I think a pulmonologist, MD, Master's of Public Health guy named Bill Rum. He's also a Minnesotan, and so he grew up from a famous family that's known for exploring the Boundary Waters canoe area. So this is this incredible wilderness country in northern Minnesota at the Canadian border and into Canada, the Quetico National Park on the Canadian side. This just high density grouping of lakes that are connected by portage trails. So you do these canoe trips where you figure out routes where you're camping for days or weeks, and you hike with the canoe on your back, and you canoe to these beautiful. I grew up on these trips, and one of my favorite places in the world. Bill Rum took him 6 decades, but he just this summer has now covered every lake in the Boundary Waters
Amazing.
and in the Quetico, connected by a portage trail. And so, another maybe sort of expression of patience and sort of slow progress. A guy who is a really accomplished and expert practicing physician in New York City who also has this really unique and beautiful, I think, connection to the wilderness, and is a witness to, over those 6 decades, to the changes in that landscape, and what that means for the impact of humans on the world, on our climate, on the air we breathe, on the water we drink, and sort of putting it in that, I think, deep perspective is
cool.
why this is my science pick of the week. So I've got a link to this article celebrating that accomplishment. And, I think the way he celebrated is he went to one of these famous outfitters, maybe on the Gunflint Trail, the lodge, sat down and drank a beer. What a great achievement and accomplishment, sort of hiding in plain sight.
I forgot to mention this comment from Riz. Sorry, I'm preaching to the choir. Nels is offering constructive. Yeah, I'm sorry. I know it's the choir, and I always hope that someone else is listening who hasn't listened before and that they'll hear this. But
Well,
you're right. I should listen to Nels.
no. Well, we need to vent too. I mean, we're on the same page. It's how we go forward is another question. Absolutely.
We have eradicated measles just by widespread vaccination in Iran, and RFK discusses if vaccination is effective or not. It's unbelievable.
And agreed.
It is unbelievable.
Absolutely right. Yeah.
Okay. All right. My pick.
Yeah.
So the other day I just stumbled. Quiet. I just stumbled onto this the other day, and it was fascinating. There's a channel on YouTube called F Stoppers. It's about photography. And the guy who does it is a professional photographer, and he made this short video. Google just made photography obsolete, and so they came out with a new AI model for not only making scenes but integrating pictures into a scene. So he does product photography. He'd take a picture of a bottle of hot sauce, and he'll photograph it in a beautiful environment. He says it takes me hours to set up. Yeah, it costs thousands of dollars for the company. And here I can do it in a minute with this new Google AI program, which is free to everyone to use. So you can upload a picture of a bottle of hot sauce and say, put this in a room with a fireplace, blah, blah, blah. And it does it perfectly. He said, why is anyone going to pay a photographer thousands of dollars now when you can do this? I mean, this is product photography he's talking about.
Right.
And, less so for people because the people still they don't look real, right? But if you don't show a face and you just show hands holding a bottle, for example, he said you can do it, and you can have the fire coming out of the bottle perfectly. And he said, I can never do that. So he said, I never thought this would happen so fast.
The advances of technology. This is fascinating. I was at a dinner with giving a seminar last year or something like that. And we had this part of maybe an offshoot of this, a debate about this, like as AI produces more art, more poetry, more music, how do you value that? Like, so currently, if you go to ChatGPT and ask it to draw something, it's kind of has a house style, it's that giveaway. But I think it's fascinating kind of philosophically and ethically. So given, obviously that AI photography, et cetera, all of this is the AI models are using all of this existing human generated work and then putting, in some cases, like as you get into copyright issues, et cetera, but even a more deeper issue. So we were just debating, and I don't know who knows what the answer is. My argument though was, I think there's still this sort of value in something that humans do, that craft. I don't know, the craft of it, the patience in it. And then it also illustrates, right, well, what's the value of this, not just for the sort of consumer, but for the producer. And, again, when we think about sort of our own careers, the science we've done, the hard work, it's not necessarily the products that I think are important and we hold up, but it's the journey along the way and what that meant both for our growth or our perspective, and then also the other people who kind of joined us or those conversations in it, and how, absolutely, I'm all in favor of using AI to poke here and there, but to not let go of the value of what we get. Yeah.
I think you can get good at AI, right? It's not that you just push a button and it's happening. You have to make a prompt and then you have to modify it. And I think there's an expertise. I mean, this guy is very good at it. You should go see and then try it because it's free. For example, I make thumbnails for all of my videos, for all my podcasts, our podcasts. And sometimes I struggle to get it right. And now I can just say, take David Baltimore in his boat and have him on a background that's uniform so it can stretch the whole thumbnail, right? And I don't have to because if you look at the thumbnail, I had to fill blue in on one side because the picture wasn't big enough, but I could have AI do that and leave David alone, right? It would just be his picture. And so, for stuff like that, I think it's very useful.
That's great. And it dovetailed beautifully from his story at the end of the podcast that you reposted about his love of fishing. And,
that's right.
So it connects that really great.
What did he say? Scientists like anything that's involved. They don't like anything that's too easy.
Anything that's absorbing. yeah, that like grabs you and you get absorbed by it. I think that was
Dixon was a big
Mm,
fisherman, right?
that's right.
And
Yeah.
once I never did that, and he said I have to show you what fishing is. So he took me to Hamilton, Montana, and we did a float on the Bitterroot
Mm,
River. And I have video of it. I have to make some kind of a
yeah, good idea.
thing of it. Him, and he just caught fish after fish after fish. Man, it was just,
I
he was
love it.
so excited. He would kiss each one and throw it back.
That's fantastic. Yeah, I'm here in fly fishing country too, and have a mentor, Jerry Kaplan, who's a retired faculty member, um, guy who grew up in the Bronx and then spent 40 years learning how to fly fish here, and then took me under his wing. And I've been absorbed on a couple of occasions, and that sort of improbability of throwing out a dry fly onto the surface of the water, and then all of a sudden a big fish comes and grabs it, and you just that has that echo of discovery, that unexpected, that almost miraculous moment where something happens that you couldn't have predicted.
So the picture I used for the Baltimore repost is a picture. He went saltwater fly fishing.
Mm-hmm,
It's
mm-hmm.
in Jamaica Bay, right here in the New
Oh, very
York
cool.
area, and he caught a huge striped
Yeah,
bass.
yeah.
But yeah, it's amazing. All right,
Agree.
that's it for TWiEVO 118. You can
Yeah.
find the show notes at microbe.tv/TWiEVO. If you like these programs, we'd love your support. I know some people supported us today. That's
Thank
great. It all goes to the operating costs of making podcasts. But if you'd like to do more, microbe.tv/contribute. And if you want to send us questions or comments or pics, I'd love to have a pic from you, TWiEVO at
Yeah.
microbe.tv. Nels LD is at cellvolution.org. I'm Blue Sky L Early Bird. Thank you, Nels.
Thanks, Vincent. Happy travels.
Oh, wait, and go over let's go over the titles here. Let me share this screen because
Yeah.
that's the easiest way to do it. So we make up titles for all these podcasts,
Oh, right. That's right. Yeah.
and so here's one. These are we make lists. Here we go. Mother of an unrelated brother,
Yeah.
sexual parasitism in ants,
Uh-huh.
polyandrous ants, domesticating males, a perfect form of male parasite, a perfect parasite, an alien genome, and a two-species super organism.
Yeah, I love it.
You
Maybe
like
we need
any
to
of
make
those?
this into a poll. Let the listeners decide what the title is.
Yeah, tell us which one you like.
That's right. That's great.
I don't know. Nels and I usually decide afterwards. But
Yeah, that's right.
we'll be back in about a month, folks.
Yeah.
Thanks for coming. I'm going to thank our moderators today. Vanity Nutrition was here, Barb Mack, and Tom Steinberg, and Les Fabi. I appreciate you all coming, and everyone else, thank you for stopping by. We'll be back with TWiEVO in about a month, and more or less the same time for office hours. I'm traveling a lot, so the next weeks of office hours, next 3 weeks, no office hours, unless I can figure something out in Greece. That's the only place I might be able to do it, but it's like a 6 hour difference. So L. Newcomb I like mother of an unrelated brother. That was the first one that Nels did. I should probably stick with that. Nevitt's like that too.
I rest my case.
All right. I'm sorry about the absence of live stream. I will do maybe in next week. I'll be in Greece. If I have a good internet in the hotel, I can do something. I don't mind staying up late. I did a TWiV from Helsinki last summer. For me, it was like 3 a.m. So
Wow.
it's possible, but just stay tuned. I will let you know. All right, Nels, I told you where Nels is from. Nels, I'm Vincent Racaniello. You can find me at microbe.tv. Music you hear on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious. Bye-bye.